All right, ladies and gentlemen, so welcome back. Today we are going to wrap up our discussion about macromolecules by talking about nucleic acids. And today's really going to be a quick crash course on nucleic acids, what they do, what their structure is, because as we get into later this school year, we'll spend a lot more time talking more in depth about this particular class of macromolecules as it relates to how your body uses the set of instructions to then actually do its job. So just to tie this back to the last video, remember we were talking about the fact that your primary structure of polypeptides, the amino acid sequence, is ultimately what determines the conformational shape of those proteins, but that primary structure is driven by your genes, driven by your genetics. And so when we define what a gene is, it's this discrete unit of hereditary information consisting of a specific nucleotide sequence of DNA. Good grief, does that sound like a textbook definition? So what does that really mean when we start unpacking that? We're talking about segments of your DNA, so stretches of DNA nucleotides, A's, T's, C's, and G's. Hopefully those sound familiar. And the order in which that code is found then corresponds to a particular set of information. So sort of think of it almost like a coded message. If I know that this code translates to this word, and I can do that enough times for enough words, I can start putting the secret message together. Another way that sometimes I describe what a gene is to kids is to think about it as chapters in your textbook, right? So your entire textbook is your overall genome, all of the DNA that your cells contain, but then we chunk it into chapters that cover a specific topic or set of topics, and your genes would be those chapters to give you a certain set of information within your overall genome. So when we look at nucleic acids as a whole, they are polymers that consist of these nucleotide monomers, so these individual pieces. So this is a true monomer-polymer structure, just like your carbohydrates and your proteins. And when we talk about nucleic acids, we're usually talking about your DNA and your RNA, your deoxyribonucleic acid and your ribonucleic acid. And those two molecules really are the driving force in the genetic information that you inherit from your parents, and then how your body actually uses that genetic information to drive its function. And so this is sort of this idea of gene expression, or what you probably learned as underclassmen is transcription and translation, protein synthesis. So DNA is your set of instructions, right? This is the actual molecule you get from your parents. But then your body has to know how to use it. Your cells have to know how to use it. And so we'll take a gene, a particular set of information, make a copy of it, and messenger RNA. That's your transcription piece, the DNA to the RNA that all happens in at least eukaryotic cells in the nucleus. That messenger RNA then gets edited. We'll talk more about that later this year. That moves out into your cytoplasm, to your ribosomes, where we actually read that particular piece of information and take that translation piece of RNA to protein. So this, another way to think about this is your giant cookbook of all cookie recipes. Today you're just going to make the recipe for chocolate chip cookies, so you just make a copy of that chocolate chip cookie recipe, take it to the kitchen, and actually make the chocolate chip cookies, or in this case, you make the polypeptide, you make the protein. So transcription is just the copying piece, right? Translation is the actual building piece, where I'm taking that written word and making a final product out of it. And we'll come back to talking about transcription and translation later this year in considerably more detail. So in order to build nucleic acids, that means they've got to have a monomer structure. And it's a string of monomers put together. The monomers of nucleic acids are nucleotides. And nucleotides are going to consist of three basic pieces. We've got your nitrogenous base, which is where those A's, T's, C's, and G's, and then in RNA, the U, or the uracil, comes from. And they have their own structure there as well. You have a pentose sugar, right? Pent means 5, so 5-carbon sugar. 4 of those carbons are here in the ring itself, and the 5th carbon's out here. And then off of that 5th carbon is a phosphate group. And the big distinction between DNA and RNA, oftentimes when we look at their nucleotides, is the pentose sugar itself. So when we talk about DNA, or deoxyribonucleic acid, that pentose sugar is deoxyribose. In RNA, it's ribonucleic acid, so ribose is the major sugar. And if you look at their structure, the only difference is deoxyribose literally is deoxygenated ribose. So here's my ribose sugar, and to make deoxyribose, we lose this oxygen off of that 2nd carbon. So there's only a hydrogen in deoxyribose. In ribose, there's a hydroxyl group. Just another term that may come up here and there. So a nucleotide contains all 3 pieces: your nitrogenous base, your sugar, your phosphate. When we talk about a nucleoside, that's just this component here. It's just the sugar and the base. All right, so it's missing the phosphate group. That term will come up here and there, so just wanted to throw that out there. So when you see it, it's a nucleotide minus the phosphate group is really what they're talking about with a nucleoside. So let's go and look at those nitrogenous bases real quick. So when we talk about these, right, you've got adenine or A, thymine or T, cytosine or C, guanine or G, and then uracil or U. Those are your 5 nitrogenous bases, that you'll find in nucleic acids. And C, T, and U are all classified as pyrimidines, or 6-membered rings, meaning they 6 carbons in the ring. And so when we look at them, really they're single-ringed bases. All right, so pyrimidines are single rings. When we look at adenine and guanine, they are a 6-membered ring fused to a 5-member, so they're a double ring. And these are your purines. So pyrimidines and purines. And when we talk about your complementary base pairs, hopefully you know A pairs with T, C pairs with G, and then when I go to RNA, A pairs with U. And if you look at it, A pairs with T or A pairs with U, G pairs with C. So you always have a purine bound to a pyrimidine. And we start talking about DNA structure later this year in more detail and how we got to figuring out what the DNA structure was. That's going to become important because it's always going to be 3 rings across. So that's going to be really important when we look at Rosalind Franklin's X-ray crystallography information and how she ended up giving Watson and Crick, I should say giving, they stole it, but the way Watson and Crick got information from her work to figure out the structure of DNA in order to make this really nice parallel ladder out of it, you needed to have 3 rings across at all times. And the only way that worked was coincidentally knowing the base pairing rules and knowing this particular structure here. So A with T or A with U, C with G, 3 rings across all the time. So we pull all of those nucleotides together to make a polynucleotide, or what most kids end up using the term nucleic acid. So not only are those bases a little different, not only are those sugars different, but typically speaking, RNA is a single-stranded molecule, although it can base pair back on itself. And then DNA is that double-stranded molecule that then twists on itself to create that sort of imaginary twisted ladder, or what we talk about as a double helix. So those adjacent nucleotides then, I'm actually going to back us up here for a sec. So when I take one nucleotide to the next, right, so this phosphate will then bond to the sugar, which will then bond to another phosphate, and so on and so forth. When we look at the bonds in the backbone here, between those sugars and phosphates, they are called phosphodiester bonds. So every macromolecule category has a very specific linkage between the monomers that build them. So when we talk carbohydrates, one monosaccharide to the next monosaccharide when they go through dehydration synthesis to make a disaccharide, and then we continue to add on monosaccharides from there. The linkage between those 2 monosaccharides is called glycosidic linkage. When you add a fatty acid chain to a glycerol, that's an ester linkage. When we add amino acid to amino acid, you get the peptide bond. to make a polypeptide. And now here in your nucleic acids, we have two more bond types. So to build the backbone of that molecule, we get these phosphodiester linkages, right? And so that's what's going to happen between the sugars and the phosphates here in this backbone. And then you also get the hydrogen bonding that happens across double-stranded DNA as well. So this becomes an interesting piece when we look at DNA replication, because in DNA you have order to the sequence. So just like we talked about that N terminus, C terminus happening in your polypeptide chain, there are ends to your DNA as well. Because those phosphodiester linkages can only happen between the 3' carbon of one sugar and the 5' phosphate, that phosphate group, I should say, that's coming off of the 5' carbon, there's order to these strands. So you'll always have a 3' end that will link all the way down to a 5' end. So here you'll have a phosphate group. This end you'll have a free sugar. And then the opposite strand goes the opposite direction. So here now I have a free phosphate group traveling all the way down to the 3' end free sugar. And so we talk about these strands running anti-parallel to each other. So they're parallel but opposite. And that's going to end up setting up some complications to the way that DNA replication happens when we get to talking about that later this year. And then, so the sugar and phosphates are in your backbone. And then in the middle of that ladder is where you get the rungs or your base pairs, A to T, C to G in DNA. And again, those are held together by those hydrogen bonds. And then you also do get some weak interactions in between the stacks of base pairs as a result of van der Waals interactions. We don't talk a whole lot about them, but your text mentions it, so I figured I'd throw that in there in case you came across that. So typically we talk the phosphodiester bonds in the backbone, hydrogen bonds between the nitrogenous base pairs as the major things that are holding these molecules together. But there are some van der Waals interactions that sort of further stabilize that DNA structure that's occurring between the stacks of base pairs as well. The strands are complementary to each other, which is really what's going to make replication possible. And so we talk about DNA replication being a semi-conservative process where we will build a new strand from the original template so that when we build that final molecule, you've got a new strand here and then part of the old strand there as well. So it's way easier to be able to say, make a copy of something if you've got something to work off of, as opposed to just being able to memorize it and then build it. So that really is an evolutionary advantage to the way that DNA works, DNA replication works, because you have the ability to be much more accurate and as a result quicker in building those molecules. And that really is a big deal when we talk about needing to build new cells for repair or even for reproduction. The efficiency of the process needs to be there as well as the accuracy. So DNA replication, we'll get into more of that process later this year, but just know that the template strand is there to serve as a way to build the new one, and that when we build those two DNA molecules, you've got one old strand with one new strand, and that's where that semi-conservative term comes from. So there's a very tight link between the DNA and the proteins, and we can sort of use this as tape measures for evolution. Something you're going to see as we continue to move through this school year is that evolution is going to be a major underlying theme to things that we talk about. We're going to constantly come back to evolution, which is why personally I like teaching you guys evolution first so that we can constantly come back and reference it. You may see in other courses, or if you talk to other students in other schools who are taking biology, they may do it in reverse. It's sort of just a personal preference. But I like kind of giving you, front loading you, and we just keep coming back to it. So no, evolution is going to be a big deal. So if you remember back, one of the big pieces of evidence that we talk about with evolution is looking at molecular data. So because DNA carries that heritable information, those genes are really necessary. I should say the genes that are very necessary to sustain life tend to be ones that don't end up with a lot of change. And so the proteins that they produce generally don't have a lot either. The more differences that we see in the DNA or in the proteins for that matter, in the way that they're sequenced and the way that they're put together, can give us some indications as to how long time has passed, how much time has passed since they have diverged from each other. So if I look at a hemoglobin protein in a human and compare that same protein to a mouse, to a gorilla, to a chicken, right? If we all carry the same hemoglobin protein, we should be able to use the number of differences that exist in that protein, and as a result, the differences in the gene that produces that hemoglobin protein, we can start putting together, piecing together how long it's been since we've diverged. So we would assume to see much fewer changes in something like a gorilla because we are much more closely related and we have diverged from them much more recently. Go back to something like maybe a chicken, and there's probably considerably more changes in those genes and in those protein sequences because we would assume that we have diverged much further back in history. So this is just a nice summary table that kind of goes over all the big pieces. What are the components? What are some examples? What are the major functions of the different macromolecules? So this might be a nice study tool to come back to in terms of major overarching pieces for this particular section. And that's a wrap on macromolecules.